Now coming to fungus. What are fungi? Fungi are achlorophyllous plants. So now how we classify them? Father of fungi or mycology is Anton de Berry, who discovered the first fungal pathogen named as Pythium dibarianum, while first Indian plant pathologist is E.J. Butler. Now first fungicide discovered was Bodex mixture or holy water, which was discovered by Millardet, which is mixture of copper sulfate and lime water. Now see, fungus is classified into two parts, false fungi and true fungi. False fungi is also known as myxomycetes or slime molds or fungal animals. This we have already discussed. There are two types, cellular and acellular fungus. In acellular, we have discussed Physarium, and in cellular we have Dictyostelium, where a specific chemical acracin is secreted, which causes the mixing of the fungi. Hence, we call them myxomycetes. Pseudoplasmodium is found in Dictyostelium, which is cellular ascomycetes, while true Plasmodium is found in acellular slime mold, which is example is Physarium. Now coming to true fungi. Why we call true fungi? Because they belong to kingdom fungi. Now see, there are four types. First is phycomycetes. Phycomycetes means algal fungi. They are having cellulosic cell wall. Example, Pythium species. This was question of CBSE 2008. Hence, we call them phycomycetes or algal fungi. Second is zygomycetes, or what we call them as zygomycetes, or we call them. Now coming to the classification of the second class, which is zygomycetes. Zygomycetes means conjugation fungi. Example, rhizophus, where heterothallium was first discovered by Blackesley. In this, we are having conjugation with the help of gametangial contact. Now, coming to the third class, which is ascomycetes. Ascus means sac fungi. In sac fungi, we have to discuss the life cycle of penicillium, and in basidiomycetes, which we call club fungi, we will discuss. We used to discuss the life cycle of agaricus species. In their life cycle, what we observe in the life cycle of both of them, what we observe? We observe first haploid thallus, which shows the plasmogamy, where we have after plasmogamy we have n plus n thallus, which go for karyogamy. Then ultimately we have 2n cell, which after iteration even again give haploid thallus. So this haploidic life cycle goes in both of them. Then now coming to the last class, which is deuteromycetes, or where we call them as fungi imperfecti. Why we call them imperfect fungi? Because sexual life stage is not found in their structure. Example, Coleotrichum falcatum. Here sexual stage is never formed. So nowadays there is report that we have observed the sexual stage of this fungus called Coleotrichum falcatum, causing the red rot disease of sugarcane. Now see, so what we observe in the entire life cycle classes of fungi is we are having from oogamy to asexual reproduction. But the primitive ones are showing oogamy, sexual reproduction, while the advanced ones are showing asexual reproduction. So what we are observing here is the reverse evolution is taking place in fungi. So that we term retrogressive evolution. Now coming to light chain. Light chain is the symbiotic association between the algae and fungi. Algae is called the phycobiont, which prepare food, and mycobiont is the fungal portion, the fungal part which absorb nutrient for that. So these symposiums are mutually engaged 24/7. So this is a very good example of symbiosis. Now see, types of light chain is on the basis of fungal partner like light chains, where we have algae together with the ascomycetes, basidiolight chain, where we have basidiomycetes together with the algae. Similarly, use of these light chains are they form pioneer species in Xerarch, and also litmus paper is prepared from them. On the other hand, we have other symbiotic association of fungi which we call mycorrhizae. This is the association of fungi with the roots of gymnosperm, gymnosperms and angiosperms living in poor nutrient soil. Now, mycorrhizae are of three types. First is ectomycorrhizae, where we are having the fungus component outside the root of gymnosperm. Second is endomycorrhizae, where we are having fungus inside the gymnosperm root. And third is vamp plant, vascular and muscular mycorrhizae, where we are having in vesicles, we are having the fungal component. These fungus used to give out the haustoria which sucks the nutrient, haustoria which sucks the nutrient, and gymnosperm angiosperm root in oligotrophic soil provide them shelter. So this symbiosis is also 24 into 7 and a very good example of mutualism.